in this part of the lecture, we will be discussing about the various other types of the vaccines, what exactly the vaccines, how exactly these vaccines are formed. Now, in the previous lecture, we discussed about what exactly the vaccines are. So, we know that the vaccines generally contains the weakened or the inactive part of a particular organism, which is called as many times referred as the antigen, which triggers the immune response within the body. So, these particular inactive parts or these particular parts are taken in. So, they are either inactivated or they are biotechnologically transformed to form certain molecules which can trigger the immune response whenever they are injected within the body. So, that is how the vaccines works in our body. Now, one of the major vaccines which are produced are called as the whole organism vaccines, which can be the inactivated or killed, or live attenuated, which are called as the avirulent strains over there. So, we just discussed about some of the live attenuated vaccines over here. Now, some of the live attenuated vaccines which were discussed in the last lecture were the tuberculosis and the typhoid. So, in this part of the lecture, we will be taking up some of the other live attenuated vaccines which are against the measles, mumps, like this one. Now, measles is basically acute illness caused by a virus of the paramyxovirus family which is there. Now, it is generally transmitted with the help of the droplets from the nose, mouth, throat of an infected person. So, we can see over here the body rashes which has developed over there. Now, the vaccine infects majorly the respiratory system over there, and it is highly contagious actually. So, it is said that lots of people die every day because of these particular attacks. Now, the first measles vaccines were developed and tested over there. So, this was a live attenuated vaccines which was developed over there. Now, this vaccine was also developed by Dr. Hillman over a period of time, where the child was treated with the measles over there. The next vaccine which came into the picture were called as the mumps vaccine. So, we can see over here the parotid glands are there, which are majorly the glands which are present in the jaw area over there. So, the child who is suffering from the mumps generally has got a swollen part in that particular area, because the parotid gland gets swelled up or inflated over there. Now, this vaccine was again developed by Dr. Hillman, and this mumps viruses was also developed with the help of the technology which was called as the live attenuated whole organism vaccines over there. Now, the mumps vaccine was given over there. So, we can see the picture where the first baby was given the mumps vaccine over there. Now, we come into the next part of the whole organism vaccines, which are called as the inactivated or the killed vaccine. Now, over here there are various vaccines which are developed, which is for the like the cholera, pertussis, plague, hepatitis A, influenza, polio, Salk vaccine, rabies, rubella, like this one. So, we will come into some of these vaccines over here. One of the first one which is is the anthrax vaccine. So, we just saw many years back there was a lots of news coming up about the anthrax as a potential agent of bioterrorism. So, these anthrax were attached to these envelopes, and whenever a person was opening this one, they were inhaled over there and they became very lethal over there. So, inhalation of these anthraxes molecules, which is basically caused by the Bacillus anthracis, it causes severe flu-like illness in that particular condition, and if it is not treated at the very particular level, it may be very lethal also. So, there is a gastrointestinal exposure which also results in the development of the ulcers in the ileum and the cecum over there. There are bacterial diarrhea which happens, sepsis happens. So, it develops into a fatal conditions over there. So, these Bacillus anthracis were developed to make a inactivated killed vaccine over here. So, the anthrax vaccine came into the picture, where these particular technique was used, and the bacterium which was the Bacillus anthracis, it was grown in the laboratory in the abnormal condition so that it get killed, and subsequently this whole organism inactivated vaccines was used in this vaccines condition. Then came the Asiatic cholera pandemic in the year 1817 to 1823, where the cholera became a very important pathogenic, killing thousands of people over there. And again this particular technique of the inactivated vaccine was utilized over here. So we can see over here how exactly the cholera vaccines was developed over there. Now the next vaccine which came into the picture was the Janus Salk. He was the scientist who developed the polio vaccine. So polio vaccines developed by him is many a times referred as the polio vaccine Salk over there. Now this was a vaccine which was initially developed for the polio over there. So this was a inactivated vaccine over there. So if we can see the picture over there, the first doses was administered in the boy over there for this particular vaccine. Now subsequently the results were announced and it was marketed over there. Now later on there were certain reports where the polio vaccinations were suspended because there were certain cases where the persons were getting the polio diseases because the viruses were getting into its normal virulent forms. The next comes the rubella vaccination. So rubella many a times referred as the German measles over there. So there is a development of the measles or the rashes on the body. Now this vaccine was also developed with the help of the technique of inactivated killed vaccines technique over there. So the rubella vaccines were developed over there with the help of the technique, and subsequently the rubella vaccines were licensed and it was utilized for the treatment of these rubella disease conditions or the rubella which was affecting the children over there. Now these vaccines, the mumps, measles and rubella, over a period of time they were combined to form a vaccine which was called as the MMR vaccine, which is now in the market. So this is a vaccine which is against the measles, mumps and rubella, which is given in a vaccine condition. So all these vaccines are combined in a single doses over there. Now there are certain vaccines which are developed from the purified macromolecules of these pathogens, which includes the diphtheria, tetanus, Haemophilus influenza, like this one, hepatitis B. So we know that certain these pathogens over a period of time, they produces certain molecules which can be purified and which can be used as a vaccine, because these macromolecules, when they are injected into the person, they elicit or trigger the immune system because they are being marked as the foreign particle. So this technique of using the purified macromolecules and subsequently inactivating them so that they cannot cause any disease and then they were inoculated as the vaccine was used over here. So these many a times are called as the toxoids over there. So we know that the bacterial toxins are produced in the bacteria, which are many a times referred as the exotoxins. Now these exotoxins, they produce many of the disease symptoms that results from the infection whenever these bacteria infect our body system. Now what happens is these exotoxins were inactivated over a period of time and they were called as the toxoids over there. So whenever these toxoids were incorporated or injected into the body, they won't cause any disease condition, but they act as a vaccine over there. So we can see the example over here, the tetanus vaccine, which is an example of the use of the toxoids over there. So these exotoxins were isolated, they were purified from the bacteria, and these exotoxins were then inactivated with the help of the formaldehyde. So there was a chemical treatment which was given to form the toxoid over there. Now these toxoid, whenever they were injected, they give the person the protection from those bacterial pathogens such as the diphtheria or the tetanus over there. So we can see over here this diagram which shows that how these bacteria generally develops or releases these exotoxins, and these exotoxins are really taken up, purified, and then they are chemically treated so that they don't cause any irritation or other reaction, but they retain the property of developing or triggering the antibodies in the person in whom these are injected. So we can see the tetanus injections which are there, the ampules which are there. So these are basically the bacterial toxins which have been inactivated with the chemical treatment, and they are referred as the toxoids over there. Now the next one comes is the Haemophilus influenza. So Haemophilus influenza is a major type of the bacteria which causes the meningitis in the children less than the age of 5 years. So as the name is suggesting, meningitis, which means that majorly the brain part is affected in these particular conditions. So we can see the bacterial hemophilia in influenza electron graph over there. Now in this hemophilia influenza, what happens is there is a capsular polysaccharide which is present on the surface. So we can see over here in the image, the red part which is showing is the polysaccharide capsules which are present on the surface of these particular things. So as these polysaccharides are taken out or they are purified, they are linked with protein carrier molecules such as the tetanus toxoid, and they are then formulated as the vaccine. So whenever these vaccines are given to the person, what happens is, as the polysaccharide capsular protein is present over there, what happens is they elicit the antibody reaction, and the antibodies are produced against the Haemophilus influenza. Now this polysaccharide protein conjugate is considered to be more immunogenic than the polysaccharide alone. So what happens is it activates the T helper cell, enable the class switching of the IgM to IgG, and then can induce the memory B cells also. The next one comes is the Streptococcus pneumoniae over there. So these bacterial polysaccharide capsules were isolated and they were developed into the vaccine condition. So we know that the streptococcal pneumonia caused the pneumonia conditions in us, majorly affecting the lungs and the other parts, respiratory parts in the body. So these bacteria were isolated, and the polysaccharide capsules which are present were isolated to develop the vaccines against them. So the vaccines which is given for the streptococcal pneumonia is called as the Pneumovax 23 over there. So Pneumovax is basically a pneumococcal vaccine polyvalent which is there. So this is a vaccine which is generally administered intramuscular or subcutaneous injections are given. Now it is basically a mixture of purified capsular polysaccharide of 23 most prevalent variants which are present, the types of streptococcus pneumonia only. That's why it is called as the Pneumovax 23 over here. Now there are certain other proteins which are produced. So these proteins are then used with the help of the recombinant technology to develop the vaccine. So the recombinant DNA technology is now also being used for the development of certain vaccines over there. Now in this, what is being done is the genes encoding the immunogenic part of that particular pathogen, which means that these are the part when they are present or administered into the body, they elicit the immune response. So these particular parts are taken in, and these genes encoding part are then cloned and expressed in the bacterial, yeast or the mammalian cell system using the recombinant DNA technology, and subsequently this produces certain, translates into proteins which can be further isolated and given up. So these are recombinant proteins which are produced, which are used as the vaccines over here. So we'll come into some of these particular vaccines which are developed through the recombinant DNA technology. Now in these recombinant DNA technology, bacterial, yeast, insect or the mammalian expression systems are majorly used for the vaccine development processes, and one of the first recombinant antigen vaccine which were approved for the human use was called as the hepatitis B vaccine. Now the hepatitis B is basically a viral infection that attacks the liver and can cause both acute and the chronic diseases over there. Now the virus is transmitted through contact, basically through the blood or the other body fluids of an infected person, and more than 7,80,000 people die every year due to the consequences and these side effects of the hepatitis B virus infection. Now the hepatitis B vaccine is able to prevent the disease condition about 95%. Now let's see how exactly the hepatitis B vaccine is being developed. Now this vaccine is called as the Recombivax HB over here. In this vaccine, what is used is, there is a part of the hepatitis B virus which is present on the surface, which is called as the HBsAg. So this is a surface antigen which is present on this particular pathogen. So this particular antigen, surface antigen, is isolated. The gene for this particular is isolated. It is cloned into a system where it can be expressed. So over here, if we see the plasmid, it is being restriction digested with the help of a particular restriction enzyme, and the part of the gene which is producing the surface antigen in the case of the hepatitis B virus is being isolated. Now both of them are ligated to form a recombinant DNA. Now this recombinant DNA is then transferred into a host cell over there, and subsequently it is being expressed over there in the host cell. Now in this expression system, as the replication, transcription, translation takes place, the proteins are produced which produces the surface antigen. So these surface antigen proteins are then isolated and purified from these host cells, and then they are packaged into the Recombivax HB vaccine condition. So whenever these particular ampules or the doses are given to the patient, they will immediately activate the antibodies or the immune system against this hepatitis B viruses, because these hepatitis B viruses also contains a surface antigens or a surface proteins which is present in this one. So it has got a immunogenicity property, so this will be elicited, and the person, as soon as he or she gets a hepatitis B infection, because there is already presence of the antibody in that particular person, the virus will be neutralized immediately and the person will be safe for the infection for that particular virus. So this is how the hepatitis B virus vaccine works. Now the next type of the vaccines are called as the multivalent subunit vaccines, which are there. Now as the name is suggesting, multivalent subunit vaccines, which means that different types of the epitopes or the antibody producing molecules are attached to a certain carrier molecule. So over here, if we see, the T cell epitope and the B cell epitopes are attached on the surface of these antibody, and now these particular carrier molecule along with the multivalent subunits are injected in the body. So what it will do is, it will elicit the immune response, because it contains the epitopes both for the T cell and the B cell. Now the T cells, as they are activated, so there will be T cytotoxic cell as well as T helper cell, while in the case of the B cell, there will be activation of the plasma cells, due to which the antibodies will produced against these particular epitopes or the antigens, and subsequently there will be production or the activation of the memory cell. So over a period of time, whenever there is an infection which happens to the person, the person will be safeguarded for that particular infection over there. The other type of the multivalent vaccines are being in the process. There are different type of the multivalent vaccines which are there. So some of them are called as the Michel form of the multivalent vaccines, some of them called as the liposomes over there. So as the name is suggesting, liposomes means there is a phospholipid layer which is present, inside which there are certain surface antigens which are have been incorporated. So the surface antigens, whenever they are injected into the body, what happens is they activate the immune system, and the immune system will go on to fight with these particular antigens over there. Now there are various subunit vaccines which have been reported over a period of time. For example, the tick-borne diseases, some of the subunit vaccines are under the clinical trials. Then the subunit vaccines against malaria, which is also against the clinical trials. Then the hookworm infection, again some of the subunit vaccines have been developed. So subunit vaccine is a very important form of vaccine which is now being developed and processed this technique. Now the next one comes is the DNA vaccines over there. Now the encoded protein of the DNA vaccines is expressed in the host in its natural form. So we know that the DNA expresses a protein with the help of the process which is called as the transcription and the translation process. Now as the DNA is present, it forms the mRNA. The mRNA formation process is called as transcription. From the mRNA, it is then translated to form the protein over there. So in this particular kind of vaccines, what happens is the DNA part is being taken up. So this DNA part is taken up, and this is then incorporated into certain expression system, and then they are injected. Now these DNA vaccines, the important part is they can induce both the humoral and the cell mediated immune system over there. Now the DNA vaccines causes a prolonged expression of the antigen which generates significant amount of immunologic memory cells over there. So all these DNA vaccines are in its clinical trial. There are lots of DNA vaccines which are in the clinical trials for various disease conditions. Now these DNA vaccines, what one of the advantages have been found is they significantly generates a immunological memory. So immunological memory is basically, it means that whenever we inject a vaccine, the immune system is activated, so the antibodies are produced. But if the person gets a infection with that particular antigen after about 3 or 4 years, there will be immunological memory cells which will be produced. So these are basically the B cells which are present in these particular persons, and as soon as the person gets infected with those particular pathogen about 3 to 4 years after the vaccination, they will immediately recognize that particular pathogen and it will neutralize it. Now by neutralizing, what happens is the immunological cells come into the picture and they neutralizes this particular vaccine pathogen over there. Now this particular image shows the pathway for the induction of the immunity by the DNA vaccines over there. So we can see over here the DNA plasmid vaccine which is there. So on the plasmid, the DNA part or the fragment which encodes for the vaccine antigen is encoded along with the promoter region, which is essentially required because this promoter region is the one which will transcribe this particular gene encoding the vaccine antigen. So as soon as these plasmid vector vaccines are injected into the muscle cells and those things, so they uses them as the expression system and they get transcribed and translated over there. So the promoter region gets activated and it transcribes the gene encoding the vaccine antigens over there, and subsequently these particular antigens are produced. Now the other thing is that they may be injected into the body, and what happens is they are being taken up by the antigen presenting cells also. They will be taken up and subsequently there will be processing over there, after which they will be showcased onto the MHC or the major histocompatibility complexes, and the immune response can be activated over there. So these are the various types. Now the various cancer vaccines are also in the developmental processes over here. Some of these processes uses the nucleic acid or some of them uses the proteins over there. Now these, what happens is whenever they are injected into the immune system, they are being taken up over there and subsequently they are being processed by the antigen processing cells, and they are being immediately recognized by the T cytotoxic cells which gets activated, and whenever there are certain tumor cells which are forming in the body, these T cells moves over there and they dislodge or neutralizes those particular overgrowth areas over there. Now we come into the last part of the lecture, which is the different types of the COVID-19 vaccines which have been developed during the COVID pandemic. So many of the vaccines we have heard over a period of time. Now in this particular types of vaccine, there are different vaccines which were developed. So the first vaccines which was developed was the DNA and the RNA type of the vaccine. So as the name is suggesting, the nucleic acid from the pathogen or the virus was taken in, and that particular thing was utilized. The next one is called as the live attenuated vaccines. So in this particular vaccines, what happens is the whole of the virus is taken up and subsequently it is attenuated, which means that they are grown in an abnormal condition over a period of time, due to which they lose their capability of disease causing, but they can induce or elicit a immune system response in these organisms per individual whenever they are injected over there. The next one comes the inactivated vaccine. So inactivated vaccine, as we discussed, are the vaccines in which the pathogen whole of the organism is taken up, the pathogen virus is taken up, and it is inactivated. So this inactivation can be with the help of the certain chemical treatment or through the help of the heat. So it depends upon these particular things. Now in this inactivated vaccines, what happens is whenever it is injected into the person, it will be recognized as a foreign particle, and the person will start developing the immune system against that particular thing. Now one of the example over here is the Covaxin over there. Now the next one comes is the subunit vaccine over there. Now the subunit vaccines, as the name is suggesting, and we have just discussed, subunit vaccines means the fragments from various parts are taken in and they are present on the surface of the particular injecting molecule. So whenever they are injected over there, they elicit the immune response. So it means that various proteins are present. So with this part, I come to the end of my lecture, in which we discussed about what exactly the vaccines is, how it functions, what are the various types and technologies used in the vaccines, like the live attenuation, inactivated one, subunit vaccines, and the various forms of the vaccine and how the development takes place. So with this note, I come to the end of my lecture. Thank you very much.